It's amazing. Like, we sort of just take plants for granted and we walk by them every single day. But how often do you actually stop and look at them and think about what kind of plants you're passing every day and how that changes throughout the year? What kind of bees or insects are landing on these plants and interacting with them? Appreciating biodiversity comes down to being aware of what's around you, even if you don't necessarily understand it.
Researchers at Whitehead Institute are seeking to push our understanding of the natural world further, to learn all we can from the stories written into the DNA of Earth's many species about how life on Earth evolved, how human beings arose, how to improve human health and well-being, and how to preserve the planet's biodiversity for the future.
When you go to the garden, the whole garden is perfect, it's beautiful. Not because there's only one species that produce many beautiful flowers. For me, it's because it has many diversity species, from early spring to deep autumn.
Fushuang Li, a research scientist in Whitehead Institute member Jinke Wang's lab, studies how pollen's protective coating evolved in many different plant lineages. He has now collected over 140 different plant species.
The whole plant kingdom, they have many species, different order, different clade, different families. Each species or each group of species, they have specific strategies to release pollen, or they have different ways to attract the pollinator.
One key to the plant kingdom's success in diversifying and spreading throughout the world is how plants protect their pollen with one of the toughest substances in the natural world, called sporopollenin.
The pollen wall structure, the sporopollenin, I think is a very important part of the plant.
The researchers determined the structure of sporopollenin, revealing what makes it so durable.
There's many different applications in the future, I believe. We can have a super strong polymer in the industry. We can use that.
Engineering plants to make sporopollenin in large amounts could even allow scientists to remove carbon from the atmosphere and trap it in a material that resists decay. The animal world, too, offers a wealth of knowledge about how to solve seemingly insurmountable problems. Amelie Roz did her PhD in Whitehead Institute member Peter Radine's lab, studying worms that regenerate. She examined both a frequently studied type of flatworm, planarians, as well as an emerging model organism, Hofstenia.
Hofstenia are able to regenerate their entire bodies like planarians are capable of doing, something that, not a lot of animals can do.
Comparing these species allowed her to explore how certain animal groups evolved the ability to regenerate.
It really is actually very exciting to be working on a new organism where you're literally writing the book on the biology in that organism.
By studying species with very different abilities than humans, scientists could unlock ways of translating the biology of those species to advance regenerative medicine techniques.
Could humans survive a spinal cord injury without paralysis? Maybe. That actually is a possibility. I think that could be generated through our study of organisms that can do nerve regeneration.
Now, Roz is studying fruit flies with Whitehead Institute member Yukiko Yamashita, looking to learn more about how flies generate new germ cells to make sperm and eggs, in the hopes of learning whether it would be possible to reprogram human cells to aid in fertility.
For every aspect of our biology, there is an organism out there that does it similarly or differently. There are exceptions to every rule, and we'll never be able to know all the different ways things can be done unless we study them.
While studying species very different from humans can unlock mysteries of biology, we can also learn more about the origins of humans by studying our closest living ancestors. In Whitehead Institute member David Page's lab, graduate student Emily Jackson is studying the evolution of primates.
I just find evolution inherently fascinating. The idea that we last shared a common ancestor with the rhesus monkey, which is one of the animals I study, 25 million years ago. Most people know the chimpanzee is our closest relative. We last shared a common ancestor about 7 million years ago. I think those numbers, it's hard to put them into scale. One of my favorite facts is that For example, mice and rats are estimated to be about 20 million years diverged. So really, the more I learn about it, the more remarkable it is to me, actually, how closely related we are to chimpanzees.
Jackson is investigating DNA samples to figure out how the X chromosome evolved to become different in monkeys, chimps, and humans.
Seeing things about how the chromosomes have changed over time can tell us what's important on the chromosome. For example, features that are shared between humans and other animals are likely to be functionally important if evolution has preserved them over millions of years.
This research led Jackson to re-examine a section of the human X chromosome that's very different among primate species.
That led me to hypothesize that maybe this region is sort of fragile or prone to deletions in humans also. And so I was able to actually confirm that by looking at DNA from thousands of men from around the world and finding that even today in human evolution, this continues to be sort of a weak point that changes rapidly and can be different between individuals. And it's not something I would have ever thought to look at without having first seen this from comparing species.
The evidence points to ongoing evolution on the human X chromosome. Uncovering the story of human evolution is only possible through the study of our closest relatives on the planet. Jackson and other researchers at Whitehead Institute do so without disturbing these vulnerable species.
It's important to study these species because in some cases there aren't other species that are sufficiently similar to people. Yeah, it would really be a tragedy to lose some of those other primate species that we share so much in common with.
In Whitehead Institute member Jonathan Weissman's lab, postdoc Richard Shih is exploring the biology of chimpanzee cells in the lab after studying chimps in their native habitats.
I've been lucky enough to participate in one summer of fieldwork where I went out to Uganda and essentially spent every day living with a community of wild chimpanzees. That gives us a lens on what the animals are like in the wild, what their behaviors are like, what their social structures are like.
Now, Shih is taking a different approach, taking a closer look at cells derived from non-invasive techniques and transformed into stem cells, so he can study chimpanzee development in the lab without impacting wild populations.
We can compare them to human stem cells, and then all the way through different developmental stages, what the differences between humans and chimpanzees are at a purely cellular level. And so, you can name 100 differences between our species that have to manifest at some point in these cells. And that's what we're looking for.
Biodiversity is precious in part because it records the story of evolution.
Biodiversity in nature is an experiment. It's a natural experiment that runs over the course of several million years. And while we're able to do experiments in the lab, we simply can't evolve a species over that kind of time scale. And so it's really all we have to work with.
One Whitehead Institute researcher is uncovering a part of evolution almost lost to history. Rebecca Povilas, a postdoc in Whitehead Institute member Marion Goering's lab, studies the world's smallest water lily, Nymphea thermarum.
If you think about a water lily, you're thinking about a pond and there's leaves on top. And then, the leaf stalks go all the way down to the bottom of the lake. Well, Nymphea thermarum doesn't actually like being in deep water very much. It actually doesn't seem that it can grow a very long leaf stalk.
While the plant is extinct in the wild, Povilas learned from other researchers how to cultivate the plant in the greenhouse in conditions that imitate the hot spring environments the plant comes from in Rwanda. It turns out that Nymphea thermarum is ideal in many ways for living in a research greenhouse.
Nymphea thermarum plants, they're small, about that big around. It's relatively quick cycling for a water lily, so it can go from seed to flowering in about 4 to 5 months. It's got a relatively small genome too.
Nymphea thermarum is important to study both because of its rarity and because it provides a unique window into plant evolution. The water lilies separated from other flowering plants early in evolutionary history, around the same time that plants first developed a nutrient-rich tissue called the endosperm in their seeds. In crop plants, this part of the seed forms the base of our global food supply.
If you're trying to understand the evolution of these reproductive characters for flowering plants, it would be really nice to know what's going on in these early diverging lineages. Sort of the modularity of how the plants build themselves, how that modularity can be altered throughout evolution. So I think that by studying early diverging lineages like water lilies, you really get a unique perspective.
Povlus's research into Nymphea thermarum underlines the importance of studying rare and declining species.
I mean, I think it's important to try and understand species before they disappear. So by studying it, you not only might learn something about how to conserve the species and maybe how to propagate it and to prevent it from going extinct, but also you just learn more about biology that way.
Researchers at the Institute are expanding the scope of species they study in order to fill in missing pieces in biological puzzles. In Whitehead Institute member Peter Redine's lab, postdoc Thomas Cook is studying differences in regenerating planarians.
I've always been interested in the naturalist perspective, just going for a walk in the woods and turning over rocks and leaves and stuff. And we do that now, when we go on collecting trips.
Not every regenerating worm has exactly the same abilities, even if they're closely related. So Cook is collecting flatworms in the wild.
For instance, there's a type of flatworm that lives in Massachusetts called Procodila fluviatilis. And it's really cool because it doesn't regenerate its head as well as the main species we use, Schmittea. And so it's a very interesting comparative study you can do. Why does this one type of flatworm that's actually fairly closely related to our favorite system not regenerate as well?
By studying why one species regenerates better than another, the researchers could reveal the biology that makes astonishing acts of self-renewal possible, in the hopes of one day applying those lessons to human health. The research underlines the benefits of following curiosity in biology.
Following your curiosity and just understanding how biology works for its own sake. Once you've done that, a lot of times, just historically, it turns out along the way, you find out how this can be used as a technology, how it can be used to better human health or produce better crops or a variety of things.
For Cook and other researchers at Whitehead Institute, studying biology in the lab has driven home the importance of preserving organisms in the wild for future generations.
Biodiversity is kind of like a library where, you may have a lot of books in a library that don't get read every day. Some get checked out more than others. But for someone doing research, it's very important to have the completeness of a large library, to know that, on that day that some question does pop into your head, you'll be able to go there and find that book dealing with that topic you're interested in. Biodiversity is kind of like that too.
Having researched primates and some of the apes, in a sense, looking at their genomes does remind you how closely related we are. So I think there's a part of it that's just almost, emotional for me at this point. It's like, look, we're actually extremely similar. I sometimes have to double check the label to make sure I'm looking at the right species because it looks almost identical in so many cases. There are many ecosystems that can't actually survive without almost all of its components. And I think you never know which components are going to be essential to an ecosystem until one is gone.
And so if we lose biodiversity, we're losing knowledge. And I think that's a really sad thing to lose. There's just so much out there that we don't know, and we don't even know we don't know it. And we're not going to learn these things until we start looking at the world around us.